Today we will study about Operon model for. This is a model Operon. We know that every gene's function, genes are responsible for the formation of protein. We have discussed that, or we are knowing that every genes, they are responsible for the synthesis of protein. But all the genes are not functional all the time in the body of an organism. It's not, for example, any gene which is responsible for the formation of any particular material. It's not that all the genes are active and continuously they are synthesizing protein. So there is a regulatory mechanism by which the functioning of a genes are controlled. For example, a gene is producing a particular material. Okay? When that material will be required by the organism, that material or that protein will be synthesized, and when this material is not required at that particular time, this protein will be not synthesized by the particular organism or by a particular gene. So there might be some mechanism by which the activities of the genes which are there, that is regulated, and that can be studied with the help of a model which is basically known as Operon. So Operon model, basically it is found in an organism, that is bacteria or E. coli. With the help of this Operon, we can study how the genes is the activity of a gene or the gene expression is regulated. For example, every gene which is there, they have different units into it. Suppose this is a stretch of a DNA. So one is the template strand and one is the non-template strand. 5' to 3', that is the non-template strand, and 3' to 5' is the template strand. Every gene has different components into it. One is promoter gene, then a structural gene, the stretch of a structural gene, and terminator gene. Promoter site is in a structural gene, and a terminator gene. This is a model of a Operon along with the promoter gene. Beside that, there is another site or a gene which is called as operator site or a operator gene. Then along with this structural gene, there is one regulatory gene. There is a gene which is called as regulatory gene, which controls the expression of this particular gene. We are studying for Operon. Operon is a model which represents the synthesis of a material, that is basically the enzymes which are essential for the metabolism of a material, which is called as lactose. This structure is found in an organism, that is E. coli bacteria. In E. coli bacteria, the genes which are responsible for the metabolism of lactose, that genes is switched on only in the presence of lactose. When lactose is there into the surrounding of E. coli, at that time the genes or the enzymes which are essential for its metabolisms are produced. When lactose is absent, at that time these enzymes are not produced. So basically these enzymes are produced with the help of a structural gene which consists of 3 parts, that is LAC-Z, LAC-Y and LAC-A. So LAC-Z is responsible for the synthesis of an enzyme which is called as beta galactosidase. LAC-Y is responsible for another enzyme which is called as lac permease, and lac A gene, which is it, that is responsible for another enzyme which is called as trans-acetylase. This beta-galactosidase enzyme is essential for the digestion of lactose. This beta-galactosidase enzymes converts the lactose into glucose plus galactose. So that's the first enzyme which is produced by LAC-Z gene, and it is essential. This enzyme is essential for the breaking down of lactose into glucose plus galactose. The second gene, which is it, that is Y, this is responsible for the formation of another enzyme which is permease, which increases the absorption of lactose within the E. coli. Okay? So this lactose which is there into the surrounding of the bacteria, that is absorbed with the help of lac permease, and the function of trans-acetylase is not very clear. So here we have to see that when this lactose is there into the surrounding, at that particular time these genes which are there, they will get switched on, and when this lactose is not there, these genes are switched off. So there is a mechanism which regulates this particular process. So as we have discussed earlier, this regulatory gene is responsible for controlling this particular process. This regulatory gene will be synthesizing from this regulatory gene a stretch of mRNA is formed, and that is responsible for the formation of a protein which is called as repressor protein. So this is a repressor protein. Repressor protein has the tendency that it binds to the operator gene. These are the two operator genes which are present here. This regulatory gene. When lactose is not there into the surrounding, at that particular time, this repressor protein, we can write down in short, this repressor protein will come and it will bind on the operator side. So when it binds to the operator side, at that particular time, the promoter gene which is there, that is blocked. The promoter gene is blocked, and when promoter gene is blocked, at that time the enzyme which is essential to catalyze this particular process, that is RNA polymerase enzyme, cannot bind to the promoter gene. It cannot bind to the promoter gene, and it cannot initiate the process of transcription. So transcription which is there, that has to be initiated by RNA polymerase. Before initiation, RNA polymerase binds to the promoter site. Since we are discussing here, when lactose is not there into the surrounding, at that particular time what will happen? The regulatory gene will synthesize a protein which is called as repressor protein. This repressor protein will bind onto the operator gene. And because this repressor protein has blocked the promoter site, RNA polymerase cannot initiate the process of transcription. But when lactose is there into the surrounding, when lactose is present into the surrounding, at that particular time, this lactose will do certain changes into the repressor protein. Because of that, some changes occurs into the repressor protein, and this repressor protein cannot bind to the operator gene. And when this repressor protein cannot bind to the operator gene, at that time this RNA polymerase gets its site to bind onto the promoter gene. At this particular instance, this RNA polymerase will come and it will bind to the promoter gene, and it will start the transcription process. Means mRNA will be synthesized, which will be responsible for the formation of three different enzymes, that is beta, galactosidase, then lac permease as well as acetylase. So all these three enzymes are synthesized in the presence of lactose. If lactose is not there at that particular time, the promoter region will be blocked by the repressor protein. So in this way we can say the gene expression is regulated. In this particular model we are seeing that the gene expression is regulated by the lactose. When the lactose is there, at that time the genes will be expressed, and when lactose is not there, the genes will be not expressed, and that is basically controlled with the help of a regulatory gene. So this model tells us how the expression of a particular gene is regulated. Thank you.